Right, so what we have here is a required practical for osmosis. So what they're using here, usually in this required practical, you use potatoes, but here we're using chicken eggs. And what we're doing is placing 5 eggs in acid for 24 hours to dissolve the eggshell. And then we're recording the mass of each one. Then we add these eggs into distilled water. So a container containing distilled water in it. That's just pure water. After about 20 minutes, we remove the eggs and we pat them dry with a paper towel and measure the mass again. So this is just like the potato experiment. Instead, we're just using chicken eggs. So first question is, a student suggested that the results for egg 4 was anomalous. Do you agree with the student? If we look at the changes in maths here, we can see we've got around 3, isn't it? So this is going from 73.5 to 77. This is around another 3. Increasing from here to here is around 3. But then if you look at number 4, it's only increasing by around 1.5. It's almost half of the other ones. Even this one is 3. So I would say it is an anomalous. So yes, we can say the change in mass is much lower than the others. Next one asks you to calculate the percentage change of mass in egg 3. That's this one here. So if you want to work out the percentage change of something, what you want to do is do the change over the initial. Change over initial and then times that by 100. So let's put the numbers we've got here. The change is between 72.4 and 75.7. So the change is going to be 75.7 minus 72.4 over the initial value. What did we start off with? We started off with 72.4. We get that, we multiply it by 100 to turn it into percentage, and the answer you should get is 4.6%. Now for part C, it's an explain question. Why did the mass of the eggs increase? So this question is all to do with osmosis, so we need to explain it in terms of osmosis. So remember, osmosis is to do with water. So what's happening if the mass of the eggs is increasing is water is moving from the water outside into the egg. And the only reason that's happening is because the water concentration outside the egg is greater than the water concentration inside the egg. So it has to pass through the partially permeable membrane of the egg, and then it makes it heavier, the mass increases. So we can say water enters by osmosis from a high concentration in the beaker to a low concentration in the egg. And then to get that third mark, mention the partially permeable membrane whenever you get a question on osmosis. Okay, so the last part is explain how the student could modify the investigation to determine the concentration of the solution inside the egg. So what they've done here is they've just used one beaker with distilled water in it. But to work out what the actual concentration is inside, we need a few more solutions. We need one with pure water, and then we need some with a different concentration of sugar in them. So you get about 5 beakers and you do the same experiment, and then you see how much the mass changes or whether the mass does even change. So something like pure water has a very high water concentration, the mass is going to increase. Then your next beaker might be 0.1 mole per dm cubed of sugar. So that's slightly lower water concentration. So the mass might still increase, but it will increase slightly less compared to the pure water. And then as you move up in concentration, the increase in mass will get less, and some of them will show a decrease in mass because at some point your water concentration in the solution is going to be lower than the water concentration in the egg, and that's going to cause water to leave the egg. Our aim in this is to find out what that concentration is. So you start off with use 5 different beakers with different sugar solutions and repeat the method. So what you're doing is you're finding the change in mass, then you're finding the percentage change, just like we did in part B. And what you're going to get is loads of different concentrations with different percentage changes. So there's a graph of percentage change against concentration. And once you plot your percentage changes, this is what you're going to see. So at the very low concentrations of sugar, you're going to have a high percentage change. The mass is going to increase. And then as you decrease, it's going to get lower and eventually it's going to turn negative. What you want is the point at which it goes from positive to negative. So you'd get a ruler and you draw a line of best fit from all your points. And this point here, where the line of best fit crosses the x-axis, that is the concentration of the egg. That's how you work it out. So we just need to explain this in words for 2 more marks. So we'll say plot a graph of percentage change against concentration. That's our second mark. And then third mark, find the concentration where the line of best fit crosses the x-axis. And there we have it, 3 marks.